Hello, learners. Every cell in your body holds nearly 2 meters of DNA. So, how does it all fit inside something you can't even see with the naked eye? The answer lies in one biology's most elegant solutions called chromosomes. In this episode, we are breaking it down. How DNA gets packed, what chromosomes actually look like, and why this structure matters more than you'd think. Welcome back to Informatics. This is Genomics Simplified Episode 3. My name is Komal. Let's get into it. Let's do a quick recap of last episode. We saw how DNA replicates itself. But here's something surprising. The DNA from just one human cell, if stretched out, would be about 2 meters long, roughly 6 feet. So, how does something that long fit inside a nucleus so tiny? That's exactly what we explore today. Long DNA gets tightly packed into chromosomes so it can fit and function properly inside our cells. Let's take a closer look at how that packaging actually works. DNA packed together in a very tight, organized way. It starts with the DNA double helix, which wraps around small proteins called histones, forming a compact structure. Here's how that folding happens step by step. First, we have the DNA double helix alone. Then it wraps around histones, forming small bead like units. These units coil together into a thicker strand called chromatin, which then denses further, folding again and again. And finally, all that folding gives us the fully packed structure we recognize as a chromosome. So, in just few steps, a long, thin strand of DNA becomes compact enough to fit perfectly inside the nucleus. Now, let's take a closer look at histone proteins, the key players that help packaging DNA. Histones are positively charged proteins, and since DNA is negatively charged, DNA naturally wraps around them. This is what allows DNA to compact and stay organized inside the nucleus. 8 histone proteins come together to form what's called a histone octamer. The DNA wraps around this core to create a structure called the nucleosome. Each histone plays its own role. H2A forms part of nucleosome core and helps to keep the DNA wrapping stable. H2B works closely with H2A to maintain the structure and control how accessible the DNA is. H3 is a co-histone involved in packaging the DNA and regulating gene activity. H4 works together with H3 to give the nucleosome its structural strength. And then there's H1, known as the linker histone, which binds to DNA between nucleosomes and helps folding everything further into higher order structures, eventually leading to chromosomes. Together, these histone acts like a support system, helping to organize DNA in a way that's both compact and functional. Now let's look at the first level of DNA packaging, the nucleosome. Think of DNA wrapping around a small spool of proteins, much like thread around a bead. This spool is made up of 8 histone proteins, 2 each of H2A, H2B, H3, and H4, together called a histone octamer. About 147 base pairs of DNA wrap around it, forming one nucleosome. Nucleosomes are connected by short stretches of linker DNA, and the fifth histone, H1, helps hold this linker region together. Under a microscope, this whole setup looks like beads strung along a thread, which is why it's called as beads on a string. This simple structure is very first step in packaging DNA to fit inside a cell. Once nucleosomes form, they don't stay separate. They fold and coil together to build a thicker structure called the chromatin fiber. Here's a simple way to picture it. DNA is like a thread and histones are like beads. Thread wrapped around one bead gives us a nucleosome. And when many of these beads come together to pack tightly, that's chromatin. So, in short, nucleosome coil and fold to form a chromatin fiber, and this is what allows a huge length of DNA to fit inside such a tiny nucleus. So far, we have seen DNA coil into chromosomes, and those nucleosomes packed into the 30 nanometer chromatin fiber. The next step is looping. This thicker fiber folds into large loops, and each loop attaches to a protein scaffold, sometimes called the nuclear scaffold. Every single loop can hold thousands of DNA base pairs. This looping helps organize the DNA, control which gene turns on or off, and get everything ready for chromosome formation when the cell divides. In short, DNA is packed like a coiled rope, then loops and anchored in place so it stays organized and ready for division. During cell division, this looped chromatin becomes highly condensed, forming the familiar egg shaped chromosome structure. This condensation protects the DNA from damage and makes sure genetic material is passed on accurately to dotted cells. But here's something many beginners get wrong. Chromosomes don't always look like this egg shape. This familiar shape is only visible after DNA replication and during cell division. Most of the time, DNA exists as loosely packed chromatin, and you can't even see it as X under regular microscope. This is actually one of the biggest misconceptions in biology. So remember, chromosomes are only condensed during cell division to protect and correctly pass on. Let's look at the basic anatomy of chromosome. Each chromosome has two identical copies called sister chromatids, joined together at the narrow region called the centromere. This centromere holds the chromatids together and also acts as the attachment point for spindle fibers during cell division. Above the centromere is the short arm called the P arm, where P stands for petit, meaning small. Below it is a long arm called the Q arm, simply named because Q comes after P in the alphabet. And at both ends of chromosome, we have telomeres, which are protective DNA caps that stop the chromosome from wearing down or fusing with other chromosomes. So here's an easy way to remember it. P is petit, meaning short, and Q follows P, meaning long. Chromosomes can be classified based on where the centromere sits, and this gives the four different shapes. First, metacentric chromosomes have the centromere right in the middle, so both arms are equal, giving a V shape during anaphase. Next, submetacentric chromosomes have the centromere slightly off center, making one arm shorter than the other, giving an L shape. Then we have acrocentric chromosome where the centromere sits near one end, so one arm is very short and the other very long, also L-shaped. And these often carry small structures called satellites on the short arm. And finally, telocentric chromosomes have the centromere right at the tip. So there's basically only one visible arm giving an I shape. Interesting fact, telocentric chromosomes are not found in humans, but they do exist in animals like mice. So remember, the position of centromere decides both the shape and the arm length of a chromosome. And this plays a big role in how chromosomes behave during cell division. So, how many chromosomes do we actually have? Every human cell contains 46 chromosomes arranged neatly into 23 pairs. Out of these, 22 pairs are called autosomes and they are responsible for most of our body functions, including growth, development, metabolism, and most of the traits we inherit from our parents. The remaining one pair is special. These are the sex chromosomes, and they decide our biological sex. Females have two X chromosomes, while males have one X and one Y. Here's a simple way to remember it. 44 autosomes plus 2 sex chromosomes gives us 46 chromosomes in total, arranged as 23 pairs. Now let's connect this something to practical. Why do chromosomes actually matter in bioinformatics? Chromosomes hold genes which carry all our genetic information and variants, which are the small differences that make each one of us unique. Together, this forms the genome, a complete set of genetic information. In bioinformatics, this data becomes really useful in several ways. First, gene mapping helps locate exactly where a gene sits on a chromosome. Second, variant detection helps identify changes like insertion, deletions, or SNPs in the DNA sequence. Third, in whole genome sequencing, we align sequencing readbacks to reference genome for analysis. Fourth, in disease research, we look at chromosomes to detect abnormalities linked to genetic disorders. And finally, comparative genomics lets compare chromosomes across different species to understand evolution. So, in short, chromosomes acts like a roadmap helping bioinformaticians analyze, interpret, and discover meaning hidden in genomic data. That's it for this episode on chromosome structure. Coming up next, in episode 4, we are diving into genes, the functional units of heredity. We will cover what genes actually are, the structure of gene, the different types of genes, how they function, and lot more. So stay tuned and don't miss the next episode of Genomics Simplified. Follow us on Instagram for bioinformatics tips, learning resources, and updates about upcoming videos. Stay curious, keep learning, and keep growing. Thank you for watching, and I'll see you in the next video.